All right, welcome back, fellow scientists. So today we are continuing our discussion of DNA replication. If you'll remember, the last time we were together, we talked about the basics of DNA replication and DNA polymerase and DNA helicase, and that's all good, but I'm pretty sure that there were some of you out there who'd be like, Mr. Leo, this is too simple. Come on, challenge me. All right, here you go. Here's your challenge. So this graphic you can find on OneNote. We used it with Mr. Anderson. We're going to use it again. And remember, Mr. Anderson taught us about the 5' and the 3' end, right? So basically, DNA is anti-parallel. 5' and 3' is just a way to say, okay, this is the top, and this is the bottom, this is the head, this is the foot, right? It's just fancy. It deals with numbering and organic chemistry and carbons and stuff like that. So this is on the sugar. This is carbon 1. This is carbon 2. This is carbon 3. So down here, carbon 3 is exposed, right? And then this is carbon 4. This is carbon 5. So then up here, carbon 5 is exposed, and and it's attached to the phosphate. And so anyways, so that's where we get 5', 3'. Don't worry about that if you don't understand that. We get into that more in AP biology. So now we're looking at why specifically DNA polymerase has to add nucleotides to the 3' end. Okay, so we're going to focus down here at the bottom of the strand, and we're going to look at this nucleotide right here, which happens to be our adenosine nucleotide. Now, you may be saying to yourself, Mr. Leo, that looks incredibly familiar. You are correct. You're remembering back to the last time we did the flip classroom, and you're looking at these 3 phosphates, and you're looking at adenosine, and you're like, ATP. But I thought that was the energy currency of the cell. I thought that was involved in photosynthesis and cellular respiration. Haha, you are correct. Guess what? It is also one of the 4 nucleotides that make up DNA. Right? So we can have ATP, adenosine triphosphate. We can have CTP, cytosine triphosphate, GTP, guanine triphosphate, and TTP, thymine triphosphate. Right? So all the nucleotides, A's and C's and T's and G's, can also have 3 phosphates and can be triphosphates. It just so happens the way that God made it is that adenosine triphosphate is also the energy currency of the cell. Whoa. That's amazing. I know. If they had a contest for most important biological molecule, ATP would definitely be a candidate because it's involved in photosynthesis, cellular respiration, anything that the cell needs energy for, it's involved in, and it's one of the 4 chemical compounds that make up your DNA that determine your traits, that give you your characteristics, that in part determine who you are. So it's pretty cool. Okay, but that's besides the point. So now we're looking at why DNA polymerase has to add nucleotides to 3' end. So here we have our little adenosine triphosphate, and it's coming in, right? And if you remember back from chapter 2 and entropy, right? What does it take to build something? You're right, it takes energy. Okay, so where does that energy come from? Well, it comes from breaking something down, right? So if we break something down like this, boop, go away, right? And then that energy is captured. We're breaking bonds and we're making bonds. So breaking those bonds give us the energy to make these bonds. So now this nucleotide is a part of this strand. And you'll notice the sugar phosphate backbone continues. The pattern is complete. So we have sugar here, we have phosphate here, we have sugar here, we have phosphate here, we have sugar here. And the base is on the inside and it's going to connect to the other base, and everything is right with the world. Okay? But now let's look at what happens if that's not the case. So if we try to add it to the 5' end, right over here, okay, so the nucleotide tries to come in, okay, and these phosphates right here, they're going to be used to attach the next nucleotide, if that actually works, which it doesn't, and we'll see, okay, because there's only one phosphate right here, so there's nothing to break down. We need this phosphate to continue the pattern, sugar phosphate, sugar phosphate, sugar phosphate, but there's no triphosphate. There's only one phosphate. It's called a monophosphate, right? So here we have CMP, cytosine monophosphate, and there's no energy to connect it, to make this bond between this phosphate and this sugar, and so that doesn't work, right? Basically, the idea is the reason why. Here, remember this. The reason why nucleotides have to be added to 3' end is because the nucleotides have to bring in their own energy, right? It's pay to play. If the nucleotide wants to be attached to the strand, then it has to bring in its own energy. It has to pay its own energy to get attached to the strand. It's not going to rely on the strand to supply its energy to connect it. it has to bring in its own energy. If it doesn't bring in its own energy, so let's look at this. So let's say that we just have AMP just kind of wandering by and, oh, hey, that looks like fun. I want to be part of the strand. And it comes in here and it's like, oh, sorry, pal. You don't have enough energy to be connected to the strand. You're not going to get to be a part of it. Oh, man, that's horrible. So then we have another ATP coming along and be like, oh, hey, that's cool. I want to be part of the strand. Boom, I have ATP. I have energy. So I am going to break this bond, attach that bond, bada boom, bada bing. It's perfect, right? So let's take that and then let's go back and let's look at this. So of course we have our base pairing rules like that, right? And then we have our DNA polymerase, which is right there. So here we have our little triphosphate, our ATP. Okay, so that's going to go right there like that. And then we're going to break off because ATP is highly reactive, so it's just gonna break off. It's just gonna go away, right? And so then we're going to paste. There we go. And we're going to bring in another. And I know that these are A's and G binds with C and T, but so this one actually works right here. So that's good. So then that's going to break off, and then we're going to make a little bond right there. And then we're going to bring in another one with its own energy. And I know A doesn't pair with A, but just go with it, right? Just bear with me here. So that's going to be like that, right? And then we can draw in our little bonds if we want to. So we can say that this phosphate attaches to this sugar and this phosphate attaches to this sugar. So now we have phosphate, sugar, phosphate, sugar, phosphate, sugar. And then here's how we represented our other two things. So that's what it would look like on our strand from before. So now let's go ahead and let's just tie this together really quickly. Let me get rid of these. And then I'll bring back our other nucleotides that we had before. Perfect. Almost perfect. Come on. There you are. Okay, cool. So now we can actually label this strand with 5' and 3'. Okay? So we know that DNA polymerase has to add nucleotides to 3' end, so that has to be 3'. We know that it's head and foot, right? So if this is the foot, then this has to be the head, so that has to be 5'. We know that DNA is anti-parallel, so if we follow this along, it's going to be anti-parallel with this strand right here. So then this strand has to be 5'. Okay? Again, we know it's anti-parallel, so if this is 5, this has to be 3. Head and foot, so we know if we go all the way back here and we started at 3, then this has to be 5. Right? And we know anti-parallel, 5 and 3. Okay, and so then we can actually check it, right? So now we have our other strand right here, and we know that it adds to the 3' end. And so then we can go, and that's going to be anti-parallel with this strand right here. So we can go, and 5 is opposite 3, perfect. And then of course that's 5, and then that's going to be opposite our 3 right here. Bada boom, bada bing, it works. God is good. All right, so last little thing that I want to show you then is the difference in DNA replication between prokaryotes and eukaryotes. Not a big difference, but still something you should be aware of. So prokaryotes, they're small, they're simple, they're bacteria, remember. So they have their DNA, their chromosome in a circle, okay, just like I have it drawn here. So prokaryotes, compared to eukaryotes, don't have a lot of DNA. So they just have one replication bubble, as you can see here, with 2 replication forks. Okay, so then they'll do their DNA replication, and then we get that, and then let's move that a little bit. There we go. And so when all the DNA is replicated, it's kind of all jumbled up, and the rings are intertwined. And then we have this cool enzyme, which you don't need to memorize, but we talk about in AP biology, called topoisomerase, which will actually nick the DNA backbone and allow the rings to be separated. And now when bacteria go through binary fission, which is their version of mitosis, now each daughter cell is going to have their own copy of their DNA. So prokaryotes, small, simple, circular chromosome, 1 replication bubble. Eukaryotes, slightly different. Eukaryotes, their chromosomes are linear. Remember our karyotypes from genetics where we numbered the chromosomes 1 through 23 and they were like propeller-shaped chromosomes lines. So each chromosome is one big long line of DNA, just like this. So eukaryotes have a lot more DNA in their cells. Humans have about 3 billion base pairs. We know that thanks to the Human Genome Project. So they're going to have in each chromosome, each of our 46 chromosomes, we're going to have multiple replication bubbles. So here I have drawn 2 replication bubbles, obviously with 2 replication forks each. So that's 4 replication forks total. Okay, and so then another kind of cool thing happens, right? So as it expands, as it gets bigger, obviously we have so they're called leading strands because they're just copied continuously. As DNA helicase unwinds, then DNA polymerase just adds, and it's no problem. Okay, we talk about this more in AP biology. You'll see it in the next video, but these strands right here present a challenge, present a problem. They can't be copied continuously because, again, we only add nucleotides to the 3' end, right? So they have to be copied in fragments. They have to leapfrog ahead and work back, and leapfrog ahead and work back. So they're going to leapfrog ahead, work back, ahead, back, ahead, back, ahead, back, ahead, back, ahead, back, ahead, back. Oop, come on. There it is. Ahead, back, ahead, back. Perfect. So those are actually called Okazaki fragments. And like I said, we talk about those more in AP biology. But even if you don't remember that, that's okay. The result is the same. We still get 2 copies of our DNA, 1 for each daughter cell. And they're semi-conservative replications. So we have our old strand, our template strand, and our new strand, our newly copied strand. Okay, so Mr. Leo, that was a lot. I wanted you to challenge me. I didn't want you to challenge me that much. I have no idea what you just said. I wasted the last 10 minutes of my life. Don't worry. It's okay. I understand. All you got to do is memorize this. If you don't understand why, that's fine. We'll get to that in AP biology. But you got to understand that DNA polymerase adds nucleotides to the 3' end. DNA replication is semi-conservative. Prokaryotes, small, simple, circular, 1 replication bubble. Eukaryotes, large, complex, linear, multiple replication bubbles. The nucleotide has to bring in its own energy, right? DNA polymerase attaches nucleotides to the 3' end. So this has been Mr. Leo. Thank you so much for watching. Today we really dove in depth into DNA replication. Believe it or not, there's more that I could have talked about, but for the sake of time and for the sake of your brains and comprehension, we'll leave that for AP biology. Thanks so much for watching, and I will see you on the flip side.